So a very warm welcome to all of you. Myself, Dr. Ritika Gautam, Assistant Professor in the Department of Chemistry at IIT Kanpur. So for today's session of CRSI 2020, our speaker is Dr. Nagma Parveen. But before I introduce our today's speaker, let me introduce everyone in the panel. So in the panel, we have Professor VK Singh, Professor Anand, Professor Raja, Dr. Dharma, and two student members. Suchismita Ghosh and Sandhya, both of them, they are PhD students. So today's session of CRSI 2020, it's even more special because we have our first women faculty to give a talk. So Dr. Nagma Parveen, she is a brilliant scientist who started her career as an assistant professor in the Department of Chemistry of IIT Kanpur recently in early 2020. She is from Rampur Hat, a small town in West Bengal, and she studied her BSc from Lady Brabourne College, Kolkata, and did her master's from IIT Madras. She further moved to Europe to do her PhD from University of Münster in Germany. Her rigorous training in the field of research also constitutes two postdoctoral experience after her PhD program. So she did her first postdoc at Chalmers University of Technology in Sweden, and her second postdoc is from Catholic University of Leuven, Belgium. So her research interest, it's mainly in the field of biophysics and physical chemistry of bionanoparticles, in particle viruses and lipid nanoparticles. Yeah. So which uses the surface sensitive and fluorescence imaging techniques, and her research lab, it will mainly focus in this field. So now I will hand over the session to Dr. Nagma and request the audience to write the questions if they have any in the chat box, and we can discuss that at the end of the session. So Dr. Nagma, you can start now.
Thank you, Ritika, for kind introduction, and thank you, Dr. Raja and Namrata, for the invitation, and CRSI for the organizing this and giving me the chance to present in this big platform. So I will share the screen now, and okay. Yeah, can you see my slide?
Yeah, we can see. You can make it full screen.
Okay. Yeah. Okay. So yeah. So this is a talk about Nobel Lecture of Physiology and Medicine, and my talk title will be Biophysical and Biochemical Perspective of Viruses. So here's a brief outline. So it is a little bit overwhelming for me to present such an important work on a, sorry, I will. Yeah. So, but I will try to, it is not really possible to summarize this huge decade of work the Nobel laureates have done in maybe 45 minutes, but that's why I planned that I will give you highlights, how they lead to this research to discovery of the hepatitis C virus. And then I will a little bit switch the gear to give a little bit more understanding, molecular level understanding of viruses and virions, so that we understand how biophysics and biochemistry can be used, because this is a CRSI talk, so I thought it will be nicer to have a chemistry perspective here. Okay, so Harvey J. Alter, Michael Houghton, and Charles M. Rice received the Nobel Prize of Physiology in 2020, and this was a discovery for hepatitis C virus. So these three scientists, they all are virologists, purely virologists. So Harvey J. Alter, he's a medical scientist, and he has been always worked in a hospital, in a medical hospital. And Michael Houghton, he's actually a biochemist by his training, but he has moved and worked in microbiology and virology. And Charles M. Rice, he is a RNA expert. And if you look in his website, he is also working with coronaviruses. And he's not only a virologist and RNA expert, but also structural biologist and biophysicist. And I also want to highlight about Harvey Alter. So he received the prize for hepatitis C virus, but he was also also the key person, so you see in this paper with Bloomberg, to discover the hepatitis B virus, which also received Nobel Prize in 1976. So he was actually the first person to visualize in immunoassay the hepatitis B virus antigen. So he has contributed both in the discovery of B and C virus of hepatitis. So it seems there's a 2 Nobel Prize for this virus, so there must be some importance of it. So hepatitis virus actually infects liver. So liver is an essential organ. It excretes all the bad stuff from our body. For example, bilirubin, that is a RBC breakdown, all the drugs we intake, that goes out from our system through the liver. So liver is a very, very important organ. And when our liver get inflamed, then this condition is called hepatitis. And typically, liver is very good in damage repairment. So it gets easily better. So it's a self-healing. But if you have a heavy alcohol consumption or autoimmune disease, or if the liver is getting infected by hepatitis A, B, C, D, or E virus, then the inflammation can go and it leads to this hepatitis disease, which can be chronic, because this virus is actually very salient. Once it infects the liver, it doesn't make a huge immune response, and that's why it's there with us for very long, until our liver get highly inflamed and results in a scarring, and which is called cirrhosis. And if it continues for 10 to 30 years, then it leads to carcinoma. So that is a cancer of the liver, and then there is no way to go back, and then only thing can be done to transplant the liver. So that's the only way to treat the patient. And here there are some numbers. You see that hepatitis B virus and hepatitis C virus cause this chronic hepatitis has caused around 300 and 150 million people worldwide in 2015 record it is. And so it is a heavy burden on us, in a medical burden, and that's why it is important that we learn about it and generate new medicine or vaccine. But the good thing about HBV is that there is already vaccine against the hepatitis B virus, and India has this universal immunization program in 2002, but it has attained very low coverage. So I think I have never received hepatitis B vaccine, and many of us actually. But today we go for hepatitis C. So I will little bit tell about the structure and how it works. So hepatitis C virus, it infects the liver cells. It's called hepatocytes, and the replication rate of this virus is very high. It replicates, so from parent to progeny virus, it's in 10 to the power 12 in number per day. But its replication is highly error prone. That means when from parent to the progeny virus generates, it's not an identical copy. And this error comes from lack of proofreading. And what is it? There are many mechanism of this kind of error. But there is an important enzyme activity called exonuclease. And this enzyme, what it does, so when so you see this virus RNA, so when the viral RNA make a copy, multiple copies, the sequence has to be read very well, right? So then only it can make a copy. But there is always some mistakes, some errors, and this enzyme tends to correct this sequence. But what happens, this HCV virus, it doesn't have this exonuclease activity. That's why when from parent to progeny virus come, it's highly get mutated. So if you compare, for example, you are hearing so much about coronavirus mutation, it's nothing close to HCV. HCV mutates so much that it becomes a quasi-species. So it's become almost a new virus. But HCV virus, also there is a difficulty, or the basic science research on HCV has been impeded because there is a lack of animal models. So there is a chimps which are used for an animal model, but there are also not that many cell culture models, and that's why there is not that much known about it in the molecular level or at a single cell level. So that's the problem with the HCV. And I think I missed to say that HCV transmit via blood, and that's why the transfusion has to be checked. The good thing that this virus is preventive, so you can prevent it, but still there is no vaccine. So I think the ideal would be if we can get the hepatitis C vaccine. So now we come to the structure of the virus. So this is the cartoon of a hepatitis C virus, and the blue one, what you see is the envelope of the virus. That is the lipid bilayer surrounding the virus, and these are the spike of the virus, which are glycoproteins. And inside the envelope, there is this orange part, what you see. It's a very important part. It's called capsid. And it's a protein shell. It's very rigid, and it protects the viral genome, in this case, the single-stranded RNA. And that's why the RNA stay protected. It doesn't get dissolved or get bad. So once this virus manages to enter in a host cell and replicate, the RNA, this RNA is nothing but messenger RNA actually. So this RNA replicates, and also it go to ribosome and transcript protein. So it creates a long polyprotein, 3000 amino acid long polyprotein, and then this long polyprotein chopped up into this constituent protein. So when this large protein breaks down and creates 3 structural proteins, one is E1 and E2 glycoprotein 2, and this core protein. So the structural protein are essential to make the structure of the virus. Without this, the virus will not form. Okay, it will not have this shape. And then there are 7 non-structural proteins, which are mainly these enzymes, which are essential for the viral to replicate. So polymerase, serine protease, and many others. And this spike, what you see, these glycoproteins, these are actually ligand of the virus. And when virus manages to enter to the host cell, if it goes to the right cell, it will bind to host receptors. So receptors are small protein or small lipids on our plasma membrane. For example, in this case, HCB binds to CD81, DC sign, LDL receptor. So these are often glycoprotein or glycolipid receptors. So, okay, so now we little bit know about the structure of HCV, and you see it's quite a complex structure. So now I will try to give 3 important papers highlight by each of the Nobel laureate, and try to summarize how the discovery of hepatitis led. So this paper in the Lancet in 1975, this actually say existence of a new virus. So what happened by 1975, it was established how to detect hepatitis B virus and hepatitis A virus. So they used to do electrophoresis and then detect these viruses exist in any person. So Harvey J. Alter, he worked in NIH hospital, and there 108 patients, they went through an open heart surgery. And for their surgery, they needed blood transfusion, and they got a donor and they received the blood. And these donors were negative according to this experiment. Okay, but still after the open heart surgery, this surgery patient, they developed, 12 of them developed hepatitis. So this was a really troubling outcome. So what Harvey J. Alter and his colleague did, they did a very thorough serological test. So they did test what antigen in their blood serum was, this patient's serum was, what were their antibody, and what they found, 4 were actually coming from this HBV. So there has been some mistake in giving, so there has been a mistake in choosing the donor. So the donors were HBV positive, and the problem was they did the test with electrophoresis, these donors, and electrophoresis is not that sensitive, and that time they established that they must do electrophoresis test and also radioimmunoassay test. So that was one conclusion. But what about the 8 other patients who unfortunately got the disease? So these 8 patients were all negative to the HBV test. So then they came to the conclusion that there is a new pathogen, they don't know whether it's a virus, which is causing this hepatitis disease, and that's why they call it non-A, non-B hepatitis. Okay, so that's why came NA and NABH. Well, so now it was established there's a new pathogen, and by then people already found out it is possibly another new virus. So Michael Houghton, So what he did, actually the problem with HCV was people who are getting infected by this non-A non-B hepatitis, they had very low level of antibody in their serum, and possibly that is because they had very low level of antigen. As I said, it's a very slow killing virus, so it was becoming very difficult to detect this new pathogen. So Houghton, what he did, he from the infected patient collected the serum, and from that extracted the RNA. And from the collected RNA, they made a library of complementary DNA. Okay, so a large library of complementary DNA, and now they have in hand this complementary DNA, and now they can use to express protein. And interestingly, the protein they expressed, it matched with the protein of this non-A non-B virus when it is infected to a chimpanzee. So this was already a proof that the cDNA library they made, they actually complement to the viral RNA. And they also did some other experiments, and from that it was confirmed that this technique, the cDNA library technique, can be used to detect this new virus. And also then they gave this name to the virus, hepatitis C virus. So this is how the name came. Well, by then people already knew there is a new virus, it is causing hepatitis. So but the problem was, as I said, this virus, the cDNA library is huge, and as I said, HCV has a high mutation rate. So there was still a problem to understand whether only this virus is causing the hepatitis. Can this virus alone cause hepatitis disease, or it needs some other viruses too, or other pathogens too? So Charles Rice, as I said, he's an expert in RNA. So what he did, he took this complementary DNA, which library was made by Houghton, and he transcribed it to RNA using RT-PCR. So he made from cDNA to tRNA, and now they have the tRNA, and then they use this tRNA to inoculate to chimpanzee and infect the chimpanzee and see whether the chimpanzees are getting this disease, hepatitis, and indeed that was the case. So it means that only this sequence of this HCV, the tRNA of HCV is enough to cause hepatitis. So this was one conclusion, but another also important output of this work was that now this tRNA is established. So now this RNA can be used to establish cell culture models. So this was also a very important contribution in the basic science. So, well, now we know that there is a new hepatitis C virus, and it can be detected using cDNA, and there is a now new model to chimps using tRNA, and we know that it can alone cause hepatitis. But at the end, of course, we want to treat the virus, right? So we want to treat the hepatitis caused by this virus. So the drug which works on this virus is a combination drug, that is a pegylated interferon and ribavirin. So when this combination of drug is administered to the patients, what happens, you see over the weeks, about 12 weeks, the RNA of this HCV virus in the blood of the patient goes rapidly down, and it stays below the detection limit. That means the patient is getting cured, because in their blood serum there is no more RNA of the virus. So you see the blue line, it stays for 72 weeks like that. So this drug works, but unfortunately about 10 to 25% of the cases this RNA level relapse, it bounce back. So actually this combination drug only works for about 50 to 60% of the patients, and some are non-responsive. This number is bad, but still in this particular paper they did this they showed the analysis, but it also depends at which stage of hepatitis this drug combination is administered. So these are also important, and still this combination of drug has been used, and it is used by many countries. There has been some new drug also, but they are specific to particular type of hepatitis C virus. Okay, so I will tell what is this interferon and what is this ribavirin. So interferon, So I will use the pen. So this interferon is a little protein, about 20 kilodalton, and it is a protein in our body, so in the host. It is actually a protein which is an innate antiviral immune system. It's a part of our immune system. So whenever we get infected by any virus, the virus, this interferon sends some signal to our immune system, and immune system becomes activated, and it releases antibody against this particular virus, whatever virus it is, and neutralize the virus. So interferon is very important. What people thought of that when hepatitis C virus attacks us, we have somehow very low immune response. So if we administered additional interferon, then it can enhance the immune effect. So that was the actually thought behind, and indeed it does. But the problem was this interferon has very it is not that stable in the blood circulation over time. So they came up with pegylation. So what they did, they pegylated particular sites using linear and branched polymer, and it indeed improved its pharmacokinetics. That means it can stay longer it can circulate longer in the blood and improve and stay there. So this is the mechanism of the interferon. What about this ribavirin? So interferon alone doesn't give very good antiviral response. Okay, so you have to add this ribavirin. So ribavirin is a prodrug, and you see the structure, and chemists will recall their studies. And it's if you close the ring here, it is nothing but a guanosine analog. Okay, so it's a ATGC. You remember for the DNA, and the G, the guanosine analog, interesting is this drug was developed for the treatment of a respiratory syncytial virus, which causes lung infection, and later on this was used for treatment of HCV. So it was not particularly developed for the HCV, but it was already there for long. I will little bit tell how this ribavirin works, very like a layman language. Actually, when a virus, as I said, when a virus infects us, its primary job will be finally to replicate its viral RNA, so replicate the RNA. So for replication, it needs to polymerize. So the RNA sequence is there. So let's say the sequence is just hypothetically A, T, U, C, and the next has to come T. And this polymerization is catalyzed by this enzyme, which is called RNA-dependent RNA polymerase. And this enzyme is enzyme of the virus. Virus has this enzyme. And of course, you need the substrate. This is the substrate, and this is the addition, the RTP, the ribonucleoside triphosphate, and that aids up. But if you add now this ribavirin, what happens? Instead of T, there will be wrong sequencing, and G will come. So I'm saying it very simplified way. So now what happens? Instead of the right sequence, the wrong sequence start to come. And in this scheme, they showed it very nicely. So when you administered So when you administer the ribavirin drug, it gets monophosphorated, diphosphorylated, triphosphorylated, and the triphosphorylated is the most it goes in this nucleophilic reaction, and you see HCV is replicating in presence of the enzyme, and when you add the drug, and then the drug causes this wrong sequencing, and that leads to defective virus formulation, and then these defective viruses are no more infectious, and we get treated by the drug. So this is also in general a mechanism of this kind of drug, this kind of nucleotide analogs. And these nucleotide analogs are not only used for HCV, as I said, it's already used for respiratory syncytial virus. It has been used for many viruses, also for HIV, and also for coronavirus. For example, remdesivir, it is also a nucleotide analog. It's in the phase 3 clinical trial, and I think it's showing some promising results. There is still some controversy. So this is the general mechanism how these kind of drugs work. Okay, so I hope I gave you a little bit of idea or some highlights how the virus was discovered and how it can be treated. Unfortunately, there is still no vaccine. I think this has to be done with little bit of policy making also, how much it is pushed to make the vaccine against the virus, because there is a treatment, so maybe that's why they are not maybe making so much push for making the vaccine against the virus. Okay, so I will now little bit give some example of some results which me and my collaborator worked together. So I can give you a little bit insight how biophysics and chemistry and biochemistry can be useful for studying in general any virus actually. So this is a simian virus which causes tumor in monkeys, and these are the structure. As you see, they are very spherical and about 50 nanometer in diameter. So the thing is, why to study a monkey virus? Yeah, so we don't care about monkeys. So actually, there is a very interesting story about it. So between 1955 and 1960, the polio vaccine, which was made, and the polio vaccines were made in the monkey's kidney, and unfortunately those monkey kidneys were infected by this virus. So what happened between these 5 years, people who got the vaccine, they also got the simian virus. And that's why there was a huge research from 1960s on that whether this virus also can cause a tumor in humans. So then whether we have any adverse effect of this virus. Fortunately, it says no. So that's good. So it only causes monkeys, the tumor. So second, a little bit I will tell about this human norovirus. This is cause a diarrhea in adults, and it's a nuisance in developed countries because a major cause of diarrhea in developed countries is this human norovirus. And I will also tell about HIV virus and how their replication can be inhibited. Okay, so when a virus manages to enter in our organ and finally reach to the host cell, the first step of its infection is to bind to the cell surface, that is the plasma membrane, and then enter. So this is the early stage of infection. So how it looks like, if you look, so the virus binds to the plasma membrane through a ligand receptor interaction. So we call this ligand the viral protein, and the molecules which are in our plasma membrane, that is receptor. And plasma membrane is full of different kind of molecules, but and different types of receptors. But the most common receptors for viruses are glycosphingolipids and glycoproteins. So I will focus on glycosphingolipid. So what is glycosphingolipid? So this is an example, GM1. This receptor is used by many viruses, toxins, bacteria, and also it is a cause. It is shown some relation with the Alzheimer disease. So GM1 is a lipid tail, as you can see, and the head group is the oligosaccharide, very specific saccharides, the sugar units. Okay, and this simian virus, it binds to this GM1. So this the simian virus ligand. So the simian virus is made of 72 viral pentamers. So this circle you see, it's a homomeric pentamer. 5 proteins are there. And this part binds to this part. So I will show a crystal structure. So in this paper, they managed to co-crystallize this VP1. This is called viral protein 1. This protein with the GM1, and this is the crystal structure. So you see 5 protein, and these little yellow ones are the GM1. And when you analyze this part very crucially, you will see that this OH, particular OH interacts with the particular amino acid sequence, and the interaction is mostly hydrogen bond or a little hydrophobic or van der Waals type of interaction. But other than this hydrogen bond interaction, you see there is also a shape matter. So how this groove of this amino acid sequence, and it has to kind of fit here. It's like a lock and key. It has to fit each other, and then only it will bind. Because people did change the GM1 structure and look at the binding affinity, and they see there is a decrease binding affinity. Only this makes the highest possible affinity. So this is about receptors. So, but we have to think whether these receptors' main job is not to bind to virus. Okay, so they are not made in our body for viruses. They have their innate functions. For example, signal transduction. For example, some of the receptors are to transport ions through the plasma membrane. And also they bind to some other non-pathogenic proteins which are important for our body. They need to be entered inside the cell. So we thought, can we use a natural protein or a peptide or a part of a protein that can also bind to these receptor and thereby inhibit this virus binding? So that was the idea. So that's what we did in this work, and we use a technique called quartz crystal microbalance with dissipation, and we also have used surface plasmon resonance, but I'm showing only QCMD data. So what we did, we made an artificial lipid bilayer with this receptor GM1. So the little black one or the GM1. And then we bound the simian virus to it. So simian virus specifically binds to it. And this is the binding kinetics of the virus. And then we come up with a little protein, CTB. It's about 5 nanometer in diameter. And that also binds to this GM1. Okay, so you see the binding kinetics here. So both binds together. But if you keep on flowing, so it's all this experiment in a fluidics. If you keep on flowing this little protein, what happens? The virus start to dissolve, release from the surface, from the lipid bilayer. So what happens? This little protein competes with the virus. And the competition happens because this little protein has much higher affinity towards GM1. So there is a competitive inhibition, and that's why it releases the bound viruses. But we only don't show that this inhibition of the virus happens. We also can now quantify, because we have the kinetics, absorption kinetics, desorption kinetics, and then we can determine the absorption isotherm. And from there, we can determine what is the binding energy. And it appeared to be the binding energy about 5 kilocalories per mole for a single contact point. So single SV40, single GM1, okay? So, well, this was for a monkey virus. Can we do the same for a human norovirus? That was the idea. So human norovirus is very, very complex, because it doesn't have only one receptor. It binds to many, many receptors, and we can group this receptor in a family, and that is called histo blood group antigens, HBGAs. As it names is blood, it relates to blood. So in our red blood cell, on the surface, we have some oligosaccharide, and it is very specific to what blood type we have, right? That's why we call A type, B type, O type. And this oligosaccharide sequence looks like this, and here are the name of the sugar units. We actually studied only these 4 types of HBGAs, and this HBGAs is not only on blood surface, but that's all in our body, in many organs. For example, in intestine, it is there. And this HBGA can be covalently linked to a lipid or a protein. This is the structure of H type, B type, A type, and Lewis A. What happens, this human norovirus we worked with binds to all 4. Okay, so we thought, what is common in them? So the common thread was this L-fucose. So the fucose definitely must have some role to bind to this human norovirus. It is not only we said, many other researchers also indicated that fucose must have some important interaction for human norovirus binding. So we thought, can we use another little protein, which fucose binding protein, that can compete, that binds to this fucose and will compete out the virus. So it can inhibit the human norovirus. So we found out a lectin, RSL lectin, that we used. And what we saw, that if we have the virus, norovirus bound to lipid bilayer, and we inject this little lectin, it actually irreversibly releases the virus. So it indeed inhibits. So we can manage inhibition of the human norovirus. And also now we are doing all quantitatively, right? So now we also have the kinetics. So now with this kinetics, we can compare the binding affinity of this virus towards these different receptors. And we did it, and here I'm showing for the 2 receptor, H type and B type. And what from the kinetic data we found, that this virus has higher affinity towards B type 1 and less affinity towards H type 1. And we have numbers also for it. So, okay, so but these all experiments are in ensemble level. So we have done this experiment on a surface about millimeter square in diameter. in area. And if we want to do the same on a single particle level, so that we can visualize them, right? So everything, if we can see it, it becomes nicer, right? So what we did, we labeled these viruses bound on a lipid bilayer. So here what you see in this image is a fluorescence image of these single viruses. So these little dots are single viruses, and we use this turf microscope. And then we, yeah, I will, yeah, okay, and then we added the little protein. So what happens to the viruses, you will see, most of the virus is released from the lipid bilayer. That means there has been inhibition because we added this competitive protein, and whatever is left, they are moving laterally on the surface and releasing from the surface. So there is a competition, and 80% of the virus is inhibited, but you still see some of the viruses stuck on the lipid bilayer. And the reason is, I'm not going to explain, but there is some kind of membrane deformation which leads the viruses to stuck. Okay, so now we see the virus in real time, and now we can also calculate how they are diffusing on the surface. We can determine their diffusion coefficient by single particle tracking, and this single particle tracking gives us information that this virus binds to how many of the receptor at a time from the diffusion coefficient calculation. And we found out majority of the virus binds to about 5 to 6 GM1, or the receptor of the virus. And if the number, so the virus is very large, it's about 50 nanometer, okay, this is 50 nanometer, and this receptors are about 1 nanometer. So the footprint of the virus is quite large, so it can bind to multiple. So we say that if it is less than 3, the virus cannot be bound, then it has to release. So this was this calculation we did from this, from the diffusion analysis. Okay, so this work, what I showed here, where we inhibit the receptor, but we can also target directly the virus. We can block the virus itself so that it is no more infectious. So we can use small molecules. With the small molecule, the problem is you need a high concentration, and maybe not all, because it's a large virus, right? So it may be not all binding sites are, for example, here you see some are not blocked, and still it can bind. But if we use some kind of a polymeric inhibitor that can kind of wrap up the virus, then maybe possibly the inhibition will be more effective. And this kind of work has been done, for example, in this paper you can look, and where they synthesize this sialated molecules, large dendrimeric molecule, and then they made a gel out of these sialated molecules, and with using the single particle turf microscopy, they indeed show the influenza virus can be inhibited, and the virus get completely wrapped up by all these dendrimers, and it's no more infectious further. So these what I show, these are the early stage of reflection. But if we want to really treat viruses, we cannot only stop them from here, but we have to stop almost every step. We have to design drugs almost every step inside the cell, so that indeed the antiviral therapy is very strong. So generally, when virus manages to enter, it goes through many steps, and it is like a unique for each virus. It's like almost like a human. They have their characteristics, and they behave differently from each other. So it is very complex to say a unique thing from one virus learning. You have to learn about individual virus for over decades, then only you can say something. So for example, this is HIV virus. It's quite a complex virus, and when it enters, it goes through the cytoplasm, and then it manages to enter our nucleus, and the viral genome actually gets integrated in our nucleus. And that's why it's such a deadly virus, because the genome becomes a part of us. And that's why a mother, when gives birth to the children, a mother with HIV positive, the children also have the HIV. So that's why it's such a ugly disease we carry out. But yeah, so that's how it is. And it is also a retrovirus. So I will tell a little part of the retrovirus, not everything. That is this little enzyme inside the, this is the envelope of the HIV, and this is this cone shape. This is the capsid, and inside the capsid there is a little enzyme called integrase. As the name says, this helps to integrate the viral genome to the host genome. So integrase looks like this. It's a dimeric protein. And I will tell how the mechanism of this enzyme is. So the mechanism is, it has two jobs. First, what it does, so this is the viral DNA. So now it processes this part of the viral DNA and make it hydroxylated, okay? So it catalyzes this hydroxylation. And then this hydroxyl group makes a nucleophilic reaction to the host DNA, and it gets inserted in the host DNA and then becomes a part of the host DNA. So the integration happens. So this is the mechanism. And now I will show a little bit of molecular mechanism. So there are many molecular mechanisms, and this is one of them. So this is the viral DNA part, you see, and this is the hydroxylated part. So this is so hydroxylated, and this part is host DNA, and this is the enzyme. And you see the enzyme makes acylation with this. So magnesium ion is very important. And then this nucleophilic reaction happens, and this viral DNA becomes a part of our host DNA. So this is how the reaction mechanism happens. So if we design a drug which can block this part of the integrase, then maybe possibly we can block this reaction to happen, and integration will not happen. So this is the active part of the of this enzyme integrase. There is already FDA approved drug called raltegravir, and this was designed actually to block this part. And this is a very good drug, and it works very good. And there has been also some analog of this drug which also works good, elbitegar, and this is another one in this paper they have shown. I think this is not yet FDA approved, but these two are FDA approved, and it is used, and it's a good drug. But increasingly there has been resistance evolving against this drug, so we need some other strategy, right? One strategy related in that field I worked, it was designing integrase inhibitor, but not really blocking the catalytic part, but in a little bit in a different way. That is, this integrase actually, viral enzyme, it interacts with a cellular protein in our cell. It's called LEDGEF. It has a long name. So yeah, so it make a complexation, and this is the crystal structure of it. So this is the integrase enzyme, and this is the LEDGEF. So this is a host cells protein, and this is a viral protein, and it make a complexation. And this complex shown to be very important for this viral DNA to tether on nucleus of the host, because before integration, this viral DNA must reach to the right site of the host DNA, right? Then only the integration happens. And if we can block this tethering, maybe possibly we will block the integration also. And this complexation, so we are blocking the tethering, and the blocking the tethering will be possible if we block this interaction. So there is in this article in Nature Chem Biology, in this article, these molecules were not new, they were there. So they made a library and looked which molecule will insert here and block this interaction. So this 6 was pinned down and tested in some immunoprecipitation reaction. But then we also did some imaging with this experiment. So what we did, we made a HIV virus, which has, of course, the integrase, of course it has, and we labeled the integrase using a fluorescent protein. So that's why it's shown a green color. And this was done in genetic engineering. So I will not go into details in the labeling part. And we did the labeling not only in one color, but 2 colors, and also made the virus in presence of this drug, this lead gene drug, okay? And then we imaged these viruses. So we did a single virus imaging. So here, this image, what you saw, these are single viruses. And we don't only do this imaging, but we also looked at how the decay of this fluorescence, so that we can learn about the fluorescence lifetime, which gives information about the interaction between 2 integrase molecules and via the FRET. And what we observed that when you have this green virus, let's say, you have a such certain value of lifetime, or that corresponds to oligomerization. And if you add this drug, the integration, this lead gene drug, the enzyme becomes more and more oligomerized. So it kind of gets multimerized, and that leads its defectiveness. And to know whether it's indeed cause a defectiveness, we have to go to the cellular level. And we did infection studies at HeLa P4 cell, and this is an image of a fixed cell, and you see the single viruses here. And I will not go the conclusion, because the conclusion is not still confirmed yet, so I will not tell much, but I just wanted to show you can do the same, the viruses you can use and infect the cell and look at the single virus in the cell at single particle level. And not only at fixed cell, but also we have done live cell imaging, where we can track single virus going. So this is the nuclear border of the cell, and it can enter from the cytosol to the nuclear border in the nucleus, and we can follow whether this enzyme is getting more and more oligomerized, and how it's changing in presence and absence of the drug. So this is one, and as I said, the conclusions are still not very clear, so we still are working on it. But one type of imaging that has given a good information, and that is fish fluorescence in situ hybridization, branched DNA fish imaging. So instead of imaging the enzyme, if we image directly the DNA, viral DNA, which is getting inserted this place, okay, if we can image that, then it would be clear proof whether this drug is working or not. So I will not go into detail how this hybridization was done. So there is a method called this branched DNA fish, which allows you to detect the integrated DNA part of the virus. And how it looks like in experiment, you image the cell. This is the transmission image of the infected cell. This is the nucleus of the cell, and you see the integrated DNA here. Very few integration occurs, and also integrated RNA. So forget about RNA now, so DNA. And you see very few integration sites, so we have to image thousands and thousands of cells. And that's why we also established an automated image analysis, so we can detect how many integration sites, and that allows us to finally quantify the effect of lead genes. Actually, in this experiment, this 6 was used. This was most effective. And the lead gene concentration dependent, you see, if you add more and more lead gene, less and less integrated viral spots you see. So that means these lead genes indeed work. But another interesting finding of the work was also that if you add the lead gene, the integration site becomes more and more inside the nucleus, not around. So it has to, the viral DNA has to go really inside of the chromosome and get integrated. Okay, so this was a little bit about the biophysics and chemistry part. So at the end, of course, we want to design vaccines, so we are all protected from any new virus coming in. So vaccine design is also a very, very important part. And the vaccine can be given intravenously, like with the blood by injection, but people are now more and more focusing on designing vaccine which are mucosal. That means it can be sprayed on your mouth or your nose, and then it goes through our mucus and gets internalized. And this when you administered a vaccine, it goes through the mucus and the mast cell, and then it go to a dendritic cell. And the dendritic cell job is to represent the antigen on its surface via MHC2, MHC1. And then there's a cascade of processes, and it's very complex. And then finally, our B cell triggers the plasma cell to release antibody when we get infected, when we really get infected by the virus, and the antibody neutralizes the virus. So then in future, if we get infected by any virus, this memory will be there, and antibody will be released, and we will be protected by this virus. So this is how in a layman language the vaccine work. So you see this part is very, very important, the dendritic cell, how it represents the antigen, the viral protein on its surface. That's the important part. So what can be done? So vaccines can be made in many, many ways. Okay, so one way which is also quite popular to make lipid nanoparticles, and conjugate a viral protein, viral antigen to it, and then administer that. So in this work, these lipoproteins were made and administered to these dendritic cells, and how well this antigen is presented. So then now this antigen, the purple proteins are presented. How much is presented will already be an indication that how good this vaccine formulation is. So this was done in this work. So we did a different, not only one type of liposome, a series of liposome. And I will give a little bit physical thing, that this composition of liposome was very important, it appeared. If you have a gel phase lipid, so gel phase lipid is often made out of saturated lipid tail and combination of cholesterol and other molecules. Then the antigen representation was much better. So as if like these gel phase lipid nanoparticles, vaccine candidates were taken up and better represented by the DC cell. So it worked better. But not only in this work, this was done, there was also a very important thing was done, that is use of adjuvant. So what is adjuvant? When we get a vaccine, it's not only we get a viral antigen or viral part of antigen or complete virus, but we also get some additional molecules. Generally, they're aluminum hydroxide or paraffin oil added. These molecules do not have any pharmaceutical property. What they do, they just somehow enhances our immune system. So addition of adjuvants propagates or helps the vaccine's immune response. So in this work, there was a adjuvant used that is called CTADD, and this is a protein, little protein, and the CTA comes from cholera toxin, because people knew when we get infected by cholera, the immune response is huge. So if we take only the catalytic part of the cholera, which is bad, but it alone cannot infect you, so but it can have the adjuvant effect. It can respond your immune system better. So in this work, this adjuvant was also added to these lipid nanoparticles, and that showed, of course, indeed a better effect. And later on, this combination of CTA-DD, lipid nanoparticles, and gel phase lipid nanoparticles were further used by our immunologist partner in the hospital, and they challenged animal models with influenza virus and used this vaccine, immunized them, and saw the effect. And indeed, these nanoparticles worked very good. So I think I'm almost at the end. So I think I said many things, but from a chemistry or biophysics perspective, I want to say it is important that we identify what are the biological questions first. And this requires that a lot of reading. We have to read a lot. For example, if we want to learn about coronavirus, if we want to contribute, then it is important that we learn how the infection mechanism of the coronavirus. We have to talk to virologists. And then with the background or with the interest in synthesis or biophysics or structural analysis, for example, you need a lot of crystal studies or electron microscopy imaging, advanced techniques like fluorescence imaging, QCMD, SPR, or if we have an interest in, let's say, data analysis or image analysis, because when we do images, we need some image analyst who can analyze this data. It's a very important part. Also theory, you saw that I showed a reaction mechanism from a theory paper, because the theory allows you to pin down the exact mechanism. Simulation and pharmacophore analysis. Pharmacophore analysis is important because nowadays there are many softwares for it, and if you have molecules, its electronic structure and steric structure can be used to understand whether this molecule can inhibit a certain type of protein-protein interactions. So with these interest and background, one can indeed answer a biological question at molecular level and relate them with basic sciences. So I will, of course, acknowledge, because all this work were done in Chalmers and Leuven with these funding sources, and in the group of Frederick Hugh and Hopkins, Hendricks, and Zager-Deviser in Leuven. And I also presented some of the work by Karin. She's the primary researcher in the work of lipid nanoparticle as vaccine candidates. Valentina, she did the immunization experiment, and Julia Janssen in establishing the branched DNA fish imaging of HIV integration. I also taken many images from Wiki and Protein Data Bank, because otherwise it's not possible to draw all images by myself. So now I'm working in IIT Kanpur, and I have a lab which is funded by SERB and IIT Kanpur Initiation Grant. So please visit Bio-Nanoparticle Lab, and if you're interested. So thank you for your kind attention.
Hi, so Raja, can we go, yeah, remove the slide share. So Nagma, thanks a lot.
I will share the slide if it is.
Yeah.
Yeah, do you see my?
Yeah. So I think. Okay. So yeah, wonderful talk. Actually, it was a very good talk, especially during this time of COVID pandemic, when we are all like studying and reading so much about virus. So here are a couple of questions that we have in the chat session. So I will start with the first one. So the first question is by Professor Verma, Sandeep Verma, Professor. Yes. So what is the effectiveness of ribavirin against various HCV genotype? The first one. Maybe you can answer this, then I will go to the next one. Yeah.
Dr. Nagma, can you unshare the slides so that we can see everybody?
Okay. Yeah. So what was the question then?
Yeah, so the first question by Professor Verma is what is the effectiveness of ribavirin against various HCV genotypes?
Yeah, so actually I looked it up. So there is one genotype, genotype 1 HCV, and that has shown not a very good response against ribavirin. And that's why there is a particular treatment. I now cannot recall whether it is a nucleotide. It is, I think it is a RDRP polymerase inhibitor, but it is a slightly different structure. But unfortunately, that particular drug is effective only for HCV genotype 1 and not others. So, still ribavirin is used for most of the genotype, but few genotypes need, particularly genotype 1 needs this other nucleotide, and I cannot now recall the name, but it is another nucleotide. Yeah.
Okay. In the same line, he has another question that what is RNA-dependent RNA polymerase the best therapeutic target for the broadest spectrum HCV intervention?
This is what has been, I think it is also not about whether polymerase inhibitor has been the best, but in general, polymerase inhibitor or protease inhibitor, in general, they have very high therapeutic index because they are the enzymes of the virus and not our host system. So they can particularly trigger those enzymes or disaffect this part of the process of the enzymes. But if we see the trend of the drugs, it seems like for also HIV-1, the polymerase, and also for other viruses, the polymerase inhibitors has been successful. But it also depends how the pharmaceutical company, what they are picking up and what they are taking on their clinical trials.
Yeah, this is the most effective target they find.
Yeah, it's a difficult question to answer actually.
Okay, so we have another question by Mr. Deepak Kumar that how effective is a vaccine if virus keeps on mutating?
Yeah, that's another thing. Of course, it is also the where is the mutation is happening. Okay, so the virus has some conserved site which very rarely get mutated, and if we make a vaccine, the antigen of the vaccine, if we make this antigen from the conserved site, that the chances are that our vaccine will work for over the years. But if we make a vaccine, maybe possibly that is also the reason the HCV vaccine making is so difficult, because the conserved site is very little and it mutates a lot. So people may have not identified the exact antigen which will keep on working for different genotype and over the different strains. But if, for example, I think for coronavirus, it is quite conserved. So if we take the conserved side, like a spike protein side, then it is possible to make the vaccine.
Okay. So, and yeah, you are getting a lot of appreciation by Professor Gurunath, Professor Patra. So you can read that later on. We have another question by Professor Patra that why COVID-19 symptoms are so diverse to the patient. Do you feel single vaccine will be effective, or one may think of personalized vaccine based on individual patients?
I think it is very difficult to make personalized vaccine. Personalized drugs, I can imagine, but personalized vaccine, because then it is no more vaccine. Because when we do vaccine trials, then it is over many, many patients. The trials are. I think maybe from 100 years from now, there will be personalized vaccine possibly. But what was the original question? It was there is a much different symptoms. Much different symptoms. Much different symptoms could be also. Means I don't know exactly, but I don't think so. Personalized vaccine will be something coming in future very soon. But yeah, personalized drug may come sometime in the future. But vaccine, because it's also a very difficult process, because vaccine is given to not infected people, not patients. It's given to healthy people. Okay, and after getting the vaccine, you can also get some disease. Okay, so there is you are, it's a big chance we are taking with something, so we have to be very careful when we make a vaccine.
Okay, so we have one more question by Mr. Badrinath Konkimala, that most antivirals have immunosuppressive effect. How about ribavirin?
No, immunosuppressive effect. Most drugs have immunosuppressive effect, but why it will be, I'm maybe not aware of. I don't think so. Most drugs will have an immunosuppressive effect. That is what I think of. But maybe I don't know the answer to it, whether it has some because generally nucleotide analogs, they don't have a huge immune response. Actually, it doesn't really. Generally, proteins, peptides. They go and they result in change in some immune response. So I will not think of a small drug molecule have a huge immune response or change in immune response.
Okay, so yeah, if anyone has any more questions, they can email you individually. But yeah, in the panel, so anyone has any question? So Chismita, Sandhya, do you guys want to ask question?
Hi Nagma, this coming back to these various symptoms, like some of them are I'm not sure, but from what I have read, a lot of those symptoms are caused by the immune response to the virus.
Yeah.
So, and of course that differs from, I think, people to people.
Yes.
So, this goes back to this one question about varied symptoms and how can those be tackled by vaccine.
Yeah.
Like you said, it's hard to get personalized vaccine, but I don't know, what are the prospects of dealing with these varied immune responses?
How to deal with this immune? Yeah, so there people are developing different type of symptoms. They are having this huge interferon response, leukin response. Yeah, it's a big thing. I'm really not an immunologist to answer this.
Yeah, no, I mean, in general, the clarity is not there yet, because for people, for example, some are given blood thinners. Some others feel joint pain. So it is all due to a body's response. So for now it is being dealt with the case-to-case basis. But there was another this comment about sort of restricting the immune response. But I don't know as a strategy how useful it will be.
Yeah, because it could also lead to some danger, right? Restricting immune response may not be. Yeah, it is important. So often people are developing this huge response quite later, or maybe some conditions. So when this huge responses come, indeed you have to, otherwise maybe we get so much inflamed, or we get high inflammation, and that could cause to death, not the coronavirus itself, but the adverse effect immune system. It's a little bit like an autoimmune disease that your body is acting against yourself. So I don't know how people, maybe people can think of how they deal with autoimmune disease and think and that direction. But truly speaking, with the coronavirus, everything new coming, it is very difficult to keep on track. But I can share my experience when we worked with this vaccine, mucosal vaccine. Truly speaking, it took 4 years or more. Before me also someone was there, and for our part was mainly making these lipid nanoparticles and conjugating different adjuvant and different, so more biophysical and their analysis. But the immunologists, when they administered to animals, every time they got different results. Because the animals also have their distinct properties, though they are grown in a very conserved way, in a very proper way, and I think they have some lineage also which they maintain, and it was very difficult to actually replicate or reproduce whatever the observations were. So thereby it took really long just for an animal model study, more than 4 years. So
yeah, no, it is challenging for sure. So
yeah,
that was a great talk, by the way. Thanks.
So Nagma, Dr. Patra, he clarified that, he meant when asking about personalized medicine based on individual patients. So he was talking mainly about the drugs.
Yeah.
So actually, I have one question before we end. So you talked about fucose binding lectin, right? Any structural specialty of these fucose that make them different compared to other ribose and ribulose for this human norovirus?
Actually, fucose is a very important, and now I cannot recall how many this OH group is there, but often when they look in the electron microscopy or in the crystal structure, maybe if you just change from one OH to C2OH, the distance of the amino acid interaction changes, or it is so the structural fits become not proper, and that leads to, so not only the hydroxyl interaction, the hydrogen bond interaction, but the structural part. But I think people have done, I cannot now recall, but people have done some variant of fucose, and they have shown some affinity, and they want to make some synthetic molecules with this variant and neutralize human norovirus. So yeah, I think as you said, other saccharide or other sugar unit will also interact, but maybe the affinity will change. That for sure.
Professor Verma actually weighed in about the comment of Dr. Nagma on restricting immune response. So he mentions restricting immune response is good if the patient is in critical stage to suppress cytokine storm. For example, he says HCQ and dexamethasone fall in that category.
Okay. Yeah, that's true.
Okay, so I think that's all for now. And if anyone has any more questions, I think they will contact you directly.
Dr. Ritika, Sandhya Singh is trying to speak, I guess, but the volume is very low.
Oh, okay.
I would like to ask a question.
Yeah.
So you talked about hepatitis A, B, C viruses. So which is more dangerous, and how can we differentiate between them by structural differences?
Yeah, so you mean hepatitis A, B, C?
Yes.
So hepatitis A, B, C, their difference come from their surface antigen. So surface antigen, so on this on the lipid bilayer, they have this glycoprotein and the core protein. So both have a different structure, and that's how you can differentiate structurally. And because they have different structure, also the antibody which we develop, that also is different, and this antibody antigen interaction strength is also quite different. So from this surface antigen and core antigen, they can be clearly differentiated.
Okay, thank you.
Ma'am, I have a question.
Yeah, go
Yes.
ahead.
Yeah. So it was a wonderful talk, very informative. So I would like to ask that you have shown the interaction of GM1 and the proteins in one slide. So what happens when after the protein interact, virus goes away? So the protein will tend to stay on the receptors, right? So it will not be able to bind other important stuff which is supposed to bind there. And another question is that how will we quantify the amount of proteins needed to remove the virus?
Yeah, so the first question is very good and very difficult at the same time, because we don't want to block important receptors forever, right? And also when we will do in the cellular or in real life, this little protein we added, they will be internalized, and the GM1 will be regenerated. So that's a bad thing about targeting this kind of receptor instead of targeting viruses. But what I can say, this has worked in most clinical drugs. For example, if you take a drug called Maraviroc, it's a HIV drug, and also this Zaminivir for the drug for influenza, they actually interfere. They don't block the receptor, but they interfere. They get stuck between the viral ligand and the receptor on the surface, and it blocks the virus to either enter or release, so it stays there. And that these two drugs has been working, and they are FDA approved. But I agree that it is not a very good strategy. Maybe targeting virus is better, using some kind of dendrimeric molecule, but such drugs has not come to the market. People always like small molecules. They don't want polymeric drugs to there has no polymeric drug even in phase clinical trials, or maybe in the initial phases. So that's one thing, and it is I agree, this is not the best suitable strategy. But maybe it can be temporarily be effective, and maybe a combination therapy could work, that you inhibit the binding for at least for some period of time, and also simultaneously you inhibit their replication inside by polymerase inhibitor or other. So this can work. And what was your second question? I forgot.
Ma'am, how would we quantify the amount of proteins needed?
That can be, for example, so we did these kinetic studies, and from the kinetics, for example, so you saw that completely the virus was completely released. So we know when we flow what is the concentration of the virus, we know the time, so we can in total, and the volume, so we don't know the amount, so we will know the amount. And if we do even the titration experiment, and if we can manage to detect, one can calculate the IC50 kind of values by simple titration using, for example, ITC method, isothermal titration calorimetry, or from the kinetics we can measure total amount was consumed. So that can be done. So you see the time, you know the concentration, you know the volume of your area, so you can calculate the amount was needed for that many viruses. So you can calculate what would be the concentration. Yeah.
Thank you, ma'am. Thank you.
Okay, so with this, we will thank Nagma for the wonderful, brilliant talk. So thank you, Nagma. So I think we will go offline, and if this video will be there on YouTube. So if anyone wants, they can go back and have a look. So